To test for the presence of proteins, two reagents are required. 10% sodium hydroxide solution and 1% copper 2-sulfate solution. Now, in the procedure for testing for the proteins, we first place the test solution in a test tube. So the solution in which you want to find out whether proteins are present or not is first placed in a test tube. So that is step number 1. Then step number 2, we add an equal amount of the 10% sodium hydroxide solution into the test tube and shake. So this is done using a dropper. So we add an equal volume of sodium hydroxide solution. Now, the standard volume for the test solution in many tests is 2 cubic centimeters. So we add 2 cubic centimeters of the test solution. Then we add an equal amount of sodium hydroxide solution into the test tube and we shake well. Then into the mixture, we add a few drops of 1% copper 2-sulfate solution, drop by drop, shaking well after each drop. So in the third step, we add the copper 2-sulfate dropwise. Now, if the solution changes to purple color, then that indicates the presence of protein. So here, if the solution turns to purple after we've added copper 2-sulfate drop by drop and shaking well after each drop, then the formation of a purple color is a positive indicator of the presence of proteins. But if the color remains light blue is a color of the copper 2-sulfate, then that is a negative test and it indicates the absence of proteins in the test solution.